So our next speaker is Chelsea Lowther. She's joining us from MGH and the Broad Institute. And Chelsea will be speaking today about balanced chromosomal rearrangements, offer insights into coding and non-coding genomic features associated with developmental disorders. Over to you. Thank you so much.
Good morning, everybody. I'm really excited to be here at my first Biology of Genomes to present our latest work on how sequencing balanced chromosomal abnormalities can provide insights into the etiology of developmental disorders. So we're going to be switching gears away from somatic mutations to focus on germline mutations. So balanced chromosomal abnormalities, or BCAs, are large cytogenetically visible rearrangements that include inversions, translocations, and insertions. De novo BCAs are significantly enriched in individuals with developmental disorders. However, we also observe them in adults with a history of normal developmental trajectories. And as it turns out, we're not very good at being able to predict the long-term clinical outcomes of BCAs based on genotype alone. And this is going to be extremely important for genomic medicine as we move forward, as whole genome sequencing becomes a first-tier diagnostic test for the assessment of developmental disorders, both that are detected prenatally as well as postnatally. So it should come as no surprise that one of the most well-characterized mechanisms of disease associated with balanced chromosomal rearrangements is the direct disruption of a known or candidate disease gene. And data from over 248 BCA carriers suggests that about 45% of cases can be explained by this mechanism. However, given that we currently do not have any cohorts of sequence-resolved BCA control carriers, we don't actually know we don't know how to quantify the disease risk attributable to direct gene disruption. And further, this leaves over 50% of cases with no known etiology, despite the fact that they all harbor one of these very large genomic perturbations that have a significant impact on genome structure and function. So altogether, this suggests that there must be alternative mechanisms of disease that are mediating the additional genetic risk for developmental disorders due to BCAs. And so data from our lab suggests that a subset of the cases that are remaining to be explained might be due to long-range positional effects. So when we take the 248 sequence-resolved breakpoints and look at their distribution, what we see is that there was one genome-wide significant region that was enriched for breakpoints beyond what would be expected by chance. And this region was particularly interesting because the vast majority of the BCAs that contributed to the signal at this region were non-coding, and all of the cases presented with an overlapping neurodevelopmental phenotype. And so when we zoom in on this region, we can see here that the BCA breakpoints are all non-recurrent. The vast majority don't disrupt a known disease gene, and they also don't disrupt any annotated cis regulatory elements. But what they do have in common is that they all disrupt a single topologically associated domain that contains a well-known neurodevelopmental gene MEF2C. So we performed qPCR on a subset of the cases and observed significant downregulation of MEF2C expression in the cases compared to the controls. And this was consistent with haploinsufficiency of this gene. So altogether, this led us to believe that we had identified a novel long-range positional effect loci, with MEF2C being the target gene, and we had this hypothesis that it might have been caused by the disruption to the 3D chromatin architecture. So we began to hypothesize that the disruption of TADs may actually represent a novel non-coding mechanism of disease that has not been well quantified in existing studies of developmental disorders. So we know that TADs contain most cis-regulatory interactions, they're stable across cell types, and highly conserved across species. And there have been a number of exceptional functional studies that have come out in the last few years demonstrating that TAD disruption can result in abnormal phenotypes in animal models. However, what we don't know is how common is this pathogenic mechanism in humans, and are there certain TADs that are especially at risk for positional effects if disrupted? So the goal of this study was threefold. We wanted to refine risk estimates due to BCA direct gene disruption, and to also identify novel non-coding loci associated with developmental disorders. We also wanted to functionally demonstrate that TAD disruption is the underlying mechanism associated with the positional effect at the 5q14.3 locus in humans. And finally, we wanted to assess whether we could identify genomic features that may define TADs at risk for positional effects if disrupted. So to address these questions, we initiated an international consortium to aggregate BCA cases and controls with whole genome sequencing data available. And so our inclusion criteria included cases ascertained for a developmental disorder or a congenital anomaly with a de novo or segregating BCA. And controls were adults without a history of an early onset phenotype, early onset pediatric phenotype, and a BCA. And I just want to mention that all of the cases and controls that were included were truly balanced at the resolution of whole genome sequencing. So these efforts have resulted in over 1,420 breakpoints from 710 individuals. And when we break this down, we have 406 cases, which represented about a 50% increase in our cohort size compared to our previous study, despite the fact that we threw out all of our complex rearrangements. And we also have, for the very first time, a cohort of controls with sequence-resolved BCA breakpoints. And so given this new data set and the fact that it was the first time we had had access to controls, the first thing we wanted to look at was what is the frequency of gene disruptions in cases, controls, and simulations. And so as expected, the vast majority of BCAs disrupt one or more genes. However, when we computationally assign all protein coding genes to one of 4 gene tiers, based on their strength of association with disease, we start to see some significant effects between cases and controls. So we see that cases with BCAs disrupt tier 1 genes significantly more than controls, but we don't see this effect for tiers 2 to 4. And this suggests that when a BCA disrupts a known disease gene, it confers about a 7-fold increased risk for a developmental disorder. So we also wanted to look at the disruption of non-coding genes, and we did not observe any significant enrichment of BCAs disrupting link RNAs or other non-coding RNAs in cases compared to controls. So the next sort of natural question was to ask, are BCA breakpoints from affected cases more closely located to known disease genes than controls? And so when we plot this data, what we see that is indeed true. There are significantly more cases with a breakpoint that are closely located to a known disease gene than controls. However, this plot is showing all of the cases that have a direct disruption of a known disease gene. So after we remove those cases and controls, the effect essentially dissipates. And so this suggests that we're not going to be able to interpret the relevance of a BCA breakpoint based on its proximity to a known disease gene. Now, in contrast, when we look at the fraction of cases with a BCA that disrupts a TAD containing a known disease gene, we see a significant enrichment for, again, our tier 1 genes when all breakpoints are included, as well as when the direct disruptions are excluded. So this provided some additional evidence to suggest that TAD disruption might be associated with pathogenic positional effects. So the next question we wanted to ask with this larger data set was, are there any TADs with an accumulation of BCA breakpoints beyond what we would expect by chance? So the first step was to remove all of the cases and controls that directly disrupted a known disease gene, since we considered these to be solved. Despite having the largest BCA cohort in the world, we're still fairly underpowered to perform direct case control comparisons. So what we did was we calculated a p-value for each TAD based on the number of cases that disrupted that TAD compared to a Poisson-Null model that was calibrated to the controls. And what we observed was 26 TADs with a false discovery rate less than 10%, 5 of which met a strict Bonferroni correction. And now these TADs are likely to represent a combination of genic effects, truly novel positional effects and other known mechanisms. So to focus in on just the positional effects, we filtered out the TADs where one of the breakpoints disrupted any gene in the genome. And what we were left with was 6 candidate pathogenic LRPE loci. And we were encouraged to see that 4 had already been reported in the literature based on case studies to represent a potential positional effect. We also identified 2 novel TADs that included 2 genes, BCL11A and BCL11B, which both code, perhaps unsurprisingly, for proteins involved in chromatin remodeling. And both of these genes have been associated with intellectual disability syndromes. And so all of the cases that disrupted these TADs presented with an intellectual disability syndrome. So overall, when we look at these 6 candidate pathogenic positional effect loci, about 7% of our case cohort disrupted one of these TADs, compared to just 3% of our controls. So this suggests that these 6 loci likely represent some of the most highly penetrant pathogenic positional effect loci in the genome. And I'm just going to step back for a minute and focus in on our favorite 5q14.3 region, because we wanted to functionally demonstrate that the disruption of 3D chromatin could be responsible for the positional effect at this locus by performing high C on some of these patient cell lines. So to date, there have only been a very small number of studies that have performed high C on cases with developmental disorders and a known structural variant. And to date, they've been unable to isolate the impact of the heterozygous S structural variant on the chromatin architecture. And this is largely due to the fact that computational workflows have not combined the phased de novo assembly of genomes with the discovery of structural variants. So we teamed up with a talented postdoc from Ayers Lieberman Aiden's lab named Olga Duchenko, who developed a phased high-C workflow to overcome this problem. And so just to briefly explain how this works, the first step is to identify SNPs and indels from the high-C data using DRAGON. We then assign each of the phased reads to one of its chromosomal homologs using the 3D DNA phaser. And then using the haploid high-C maps, we then apply the juicebox assembly tools to discover structural variants. And so this really allows us to reconstruct the end-to-end sequence for each chromosomal homolog that reflects sort of the true contact pattern while also taking into consideration the local structural variation. So we applied this pipeline to 5 cases with non-coding BCAs disrupting the TAD containing MEF2C, including 1 translocation, 1 inversion, and 3 complex cases. And we were very encouraged to see that the high C recapitulated about 96% of the breakpoints that we had originally identified with whole genome sequencing. So it was able to identify the correct structural variation and put it in the correct orientation. So I'm just going to show you the data from one of our most complex cases, which included over 40 different breakpoints. So this is an example of high C data mapped to the HG19 reference of the human genome, and I'm just showing you what it looks like for the 5 megabases region around the 5q14.3. When we take the phased high C data from our case and map it back to the reference genome, what you basically see are all these cuts. And basically that is identifying the structural rearrangement. So you know there's something going on here, but you don't know what that rearrangement, what kind of impact it's having on the actual architecture. So after we apply the phased high C data back to basically the personalized rearranged chromosome, you're able to actually see what the resulting 3D architecture looks like after taking the rearrangement into account. And so you can see here that there are a number of boundaries that don't exist in the original map, suggesting that the rearrangement is creating new boundaries. So next, we just wanted to see if there was a reduced frequency of contact between the MEF2C promoter and its annotated enhancers. So we took 16 neuronal enhancer annotations and compared the contact frequency between the intact chromosome and the rearranged allele. And what we observed was a significant reduction in contact frequency. And when we looked at the actual high C maps, it seemed like this reduction in frequency happened through two separate mechanisms. Either the BCA was increasing the distance between the promoter and the enhancer so far that they could no longer contact, or the enhancers stayed in the same linear distance to the promoter, but the creation of a novel boundary prevented them from contacting. And so now we're carrying out additional studies on other BCAs to determine if these are the only two mechanisms or if there are additional ones that we can discover. So I'm just going to finish up really quickly with one last analysis. What I've previously shown is that there is an enrichment of BCA breakpoints that disrupt TADs housing known developmental disorder genes. However, there are still 40% of cases that have the exact same feature. So there must be additional features that characterize TADs that are susceptible to known positional effects. So to see if we could identify some of these features, the first thing we did was we compiled a list of 54 coding and non-coding genomic features and annotated all autosomal TADs in the genome. We took the TAD data and defined our training data, which included 45 TADs that were disrupted by 2 cases and no controls, and 261 TADs were our negative training data, which were not disrupted by any cases, but just disrupted by 1 control. We performed a univariate analysis and identified 18 features that were nominally associated with either enrichment in cases or enrichment in control TADs. But given that we know most of these features are correlated, we applied a penalized regression, which ultimately identified 6 features that were independently associated with TADs disrupted exclusively by cases. And so these included ultra-conserved elements, VISTA enhancers, transposon-free regions, and our favorite developmental disorder genes. So to just make sure that to evaluate our model, the first thing we did was we defined an independent set of 372 TADs and applied our model to see how well it was predicting case or control outcome. And we observed that it was doing a fairly good job, better than just overlapping our TADs with any single individual annotation. And a second thing we wanted to do was to define a pathogenicity cutoff score. So we applied our model to all TADs in the genome and looked for the point at which there was an enrichment of cases versus controls. And then we went to the literature and identified 17 known positional effect loci that were not included in our training data. And what we observed is that 75% of these previously associated loci were ranked above this threshold that we had defined. And this was significantly a significant enrichment compared to all other TADs in the genome. So just to summarize what I've presented today, we know that the functional impact of BCAs involves the reorganization of the 3D genome. We've shown that TAD disruption represents an important source of non-coding risk for developmental disorders and may explain an additional 7% of developmental disorder cases with a BCA. We have preliminary data to suggest that genomic content of the TADs are also important for determining disease risk, but our models are not powerful enough yet to discriminate cases from controls, and so we're going to need larger data sets and more sophisticated statistical models before we could ever use something like this for the clinical interpretation of balanced chromosomal abnormalities. And with that, I just want to thank everybody in the lab and all of our collaborators, and I'm happy to take any questions.
We have time for one short question, if there's one from the audience. If not, I have 1.5 questions. The one is whether there is precise breakpoint recurrence at all that might allude to a mechanism that causes these events?
No, in our entire data set, there wasn't a single breakpoint that was recurrent.
And the second is a very obvious question about long-read technologies and whether you can use them to elucidate the more complex events.
Yes, so about 10% of the BCAs cannot be identified with short-read whole genome sequencing. So right now what we've done is we've taken those cases that we couldn't resolve and we're doing long-read whole genome sequencing to see if we can resolve them, because it's going to skew our results a little bit.
Okay, thank you so much, Chelsea.